let's begin part 2 of biochemistry with DNA repair. So before saying DNA repair, you need to see two things. One is depurination, deamination. According to name, you can see depurination means remove the purines like adenine and guanine. Deamination means loss of amines. So because of loss of amines, you can't convert cytosine into uracil. Okay. This is just one of the example. Now coming to DNA repair. DNA repair, you can repair the double stranded DNA which is broke, and also you can repair the single stranded DNA which are broke. So let us see one by one. The first one is double stranded DNA. They are divided into non-homologous end joining and homologous recombination. Non-homologous end joining, you can see here the double-stranded DNA is broke and it is now joined. Non-homologous means they are not similar. There are of different types of DNA, but they are joined. But this type is having high chance of error, so it is highly error prone. Next, coming to homologous combination. Homologous combination means you need sister chromosomes. Sister chromosomes are required here. Then only they can unite again. You can see here this pink one is repair. It's undergoing repair in the presence of blue one, which is a homologous chromosome, homologous DNA. So homologous recombination means the same sister chromosome are required, and this kind of condition, this homologous repair, is absent in female cancers, especially breast and ovarian cancers, which are associated with BRCA1 and BRCA2 mutations, and also in Fanconi anemia, which is inherited aplastic anemia. Here also there is no homologous recombination present. Now coming to single stranded repair, the first one comes the nucleotide excision repair. Actually here, according to name, you can see nucleotide excision. We remove one nucleotide and we place a proper new nucleotide. So let us see the process. The first one is, you can see the pyrimidine dimer. The pyrimidines, if you remember, they are cytosine, uracil, thymine. So here the pyrimidine is TT, that is dimer. So this dimer, because of some UV exposure or something, it has got damaged, and now we need to remove it and add a new kind of strand of DNA. So to remove that, we have an enzyme called endonuclease. Okay, this removes the damaged segment, and now our DNA polymerase, which is important enzyme to form a new strand, which comes into place and forms a new strand. And now this new one should be attached to the old one by means of some gum. That gum is nothing but given by DNA ligase. So by all these enzymes, a new strand is formed. This is nucleotide excision repair, and this undergoes in G1 phase of cell cycle, and there is a clinical association with this, that is scleroderma pigmentosum. In the name itself, you can say sclero. Sclero means very dry. Derma means related to skin. So dry skin, pigmentosum means there is some photosensitivity involved here, and so there's high risk of skin cancer. This is seen in childhood only, that is childhood disorder. So here the problem is this nucleotide excision repair can't occur here. So because of that, you are getting that skin problem. Now coming to base excision repair. According to the name, you can see base excision. We remove one base. So here, for example, the deaminated cytosol is nothing but uracil. So now we want to remove this. So after removing this, there's a empty AP site. So removing this is done by glycosylate. Okay, so here it is removed by endonucleases. Here in base excision, it is removed by glycosylate. So this glycosylate removes the base at the AP site. AP site is nothing but apurinic or apyrimidic site. In the name itself, you can see apurinic, no purine, apyrimidine, no pyridine. So these both are not present, and very important is there are some other two enzymes which are helpful to remove this U out of this. That is, at the 5' end you have endonucleases, at the 3' end you have lyase enzyme. So these both are also helpful to remove this base out. So now you have removed this base out. You can see as here it is mentioned endonuclease, lyase, endonuclease at 5' end, lyase at 3' end. They remove the backbone. Now we add the new one, that is, you can see C to G, because C and G will have triple bond between them. So this new C is now added, and it is by polymerase beta. Especially here, polymerase beta fills the gap, and DNA ligase, as usual, it will seal it. So now coming to mismatch repair, and one more thing, this base excision repair will undergo in all phases of cell cycle. Now coming to mismatch repair. According to the name, it is a mismatch between A and G, because A should bind with the T, right? So it is a mismatch. It finds the mismatch, and we will remove the mismatch. Okay, so this nucleotide is removed, and new one is formed, and it is again resealed. So the same process here, this is a mismatch repair. It is in S and G2 phase. This is very important for microsatellite stability. So if this mismatch repair is not present, that will lead to microsatellite instability, and this can be seen in HNPCC, that is hereditary non-polyposis colorectal cancer. So in colorectal cancer, this mismatch repair system is not working. So because of that, there is Lynch syndrome and leading to colon cancer. Now let's begin with mutations of DNA. So mutations of DNA divided two types: germline mutation, somatic mutation. Germline, you can see it is like in the mother, like it can be transmitted to the offspring, whereas somatic mutation, it is not transmitted to the offspring. So mutations are of different types, like single nucleotide. There are single nucleotide mutations and some other big type of other mutations. So let us see the mutations one by one. Before that, you need to see the two types of single nucleotide point mutations, that is transition and transversion. Transition is most common. Transition occurs between the same type, like from purine to purine, like there is a transition of A to G, from pyrimidine to pyrimidine, like C to T. Okay, transversion is transverse, like it is purine to pyrimidine, pyrimidine to purine, so it is of opposite type. Now coming to single nucleotide substitutions. So silent mutation, here in the according to diagram you can learn it very easily. Silent mutation is that you change one kind of amino acid, same type of an amino acid, which codes actually for the same codon. For example, here instead of A you change some P. Even though you change it for P, it will code for the same type of amino acid, like it has same codon. Okay, especially this occurs in third position. You can see codes for the same amino acid often involves third position of codon, and this is also called TRNA wobble. Next, coming to missense mutation. Missense mutation is there's a change in amino acid. For example, you can take sickle cell anemia, where there is substitution of glutamic acid with valine. So here there will be a change. In sickle cell, you will see sixth codon change. Next, coming to nonsense mutation. Here you can remember it by stop the nonsense, because early only, in the early stage only, the stop codons will come. You know, stop codons are you go away, you are away, you are gone, mnemonic, UGA, UAA, UAG. So these are the stop codons. They appear early, then that is called as nonsense mutation. Okay, and because of this, it will lead to formation of non-functional protein, and it will terminate the translation process very quickly. Now coming to other types of mutations: frame shift mutation, splice site mutation. So frame shift, according to the name, you can see the complete frame of the codon will be changed here. Either there will be deletion, or that is if you can remove some, you can see frame shift deletion, you are removing some, or there can be addition, that is insertion of some kind of nucleotide. So because of this, there will be all misreading of the nucleotides, and the protein may be either shorter or longer. For example, if the early stop codon will be early, then there will be small protein, and if the stop codon is delayed, then it will be long protein. So this kind you can see in Duchenne muscular dystrophy, Tay-Sachs disease. So this is leading to change in the DNA size because of this frame shift mutation. But in case of point mutation, you will have no size change of DNA. Okay, you can't find any change on DNA size, but in frame shift you will change the DNA size. Next, coming to splice site mutation. This is actually seen because of retained intron. Actually, intron should be removed when you convert mRNA into mature mRNA. You can see here. Actually, introns should be removed, but if introns are still present, then that will lead to formation of some abnormal protein. So because of that, that will lead to impaired or altered function of that protein. Example is in rare cases of cancer, dementia, epilepsy, some types of beta thalassemia, Gaucher disease, Marfan syndrome, all this. And cystic fibrosis. In cystic fibrosis, you can see the most common mutation is delta F508. There is a deletion of 3 DNA bases in cystic fibrosis. There is one more thing: slipped strand mispairing. Slipped strand mispairing occurs in repeated nucleotide sequences. So these are some kind of mutations in DNA. Now let us see about lac operon. So lac operon, what is this? Is first you need to understand about this repressor protein. So before that you need to see there is a promoter, operator, okay, and a repressor. So repressor is inactivated whenever there is lactose. That thing first you need to remember in your brain. Whenever there is a lactose, repressor will be inhibited. So if repressor is inhibited, the operator is activated and it will activate the promoter, like that you can think. But whenever the repressor protein is not with lactose, then repressor will activate and it will go and wind around the operator and it will block the operator. So when the operator is blocked, of course the promoter will be blocked and the system will be blocked. Okay, so first we need to understand the system is whenever there is no glucose, what happens is this ATP in the presence of adenylyl cyclase, it will convert into cyclic KMP, and this cyclic KMP will activate the cap protein. This binds a cap site and induces the transcription. So when transcription comes, you know this lacoperon, this structure is called lacoperon. That is when there is no glucose, whenever there is lactose, this process goes on. That is the promoter region is activated and that will lead to transcription. So transcription factor is activated whenever there is lactose. But if glucose is there, then this adenylyl cyclase will be inhibited and all this process will be inhibited. Repressor protein will be activated. But when lactose is present, repressor protein will be inactivated. Okay, you can see here high lactose unbinds repressor protein from operator side and increase transcription. So here there are different cases to understand. First you can see whenever there is low glucose but lactose is still available, then what happens, the lac genes are strongly expressed because there is low glucose, there is more lactose. But whenever there is high glucose and lactose is unavailable, clearly you can understand glucose is an inhibitor, so this lac gene is not expressed. And whenever there is low glucose and lactose is unavailable, very low glucose, lactose is unavailable, of course lac genes are not expressed. High glucose, lactose is still available but there is more glucose which is dominating it, so because of that this system will be inhibited. So very low basal expression. So this is about lacoperon which we need to know. Next thing what we need to know is about trinucleotide repeat disorders. In the name itself you can see three nucleotides are repeating in case of Fragile X syndrome, Friedreich ataxia, Huntington disease, myotonic dystrophy, and microsatellite instability is seen in short repeated segments of DNA which is seen in colon cancer. We already discussed this. Now let's come to the main part, that is how the mRNA is formed. So first one we need to know some regulation of gene expressions. So let us see one by one. The first one is the promoter region. You can see this is a site where actually the RNA polymerase binds. Here the RNA polymerase 2 binds and some other multiple transcription factor can bind to this site, which is especially AT rich site. This is also having TATA and CAT boxes. These are called as promoter regions. You can see here. Here the DNA polymerase 2 can bind. It is differing in both eukaryote and prokaryote. So if there is some mutation for this promoter region, that will lead to decrease in gene transcription, of course. Next, coming to enhancer. You can see there are enhancer and silencers, that is which can either enhance or they decrease the transcription. So enhancers, in the name itself you can see they can increase the transcription by activating actively binding to the RNA polymerase so that it will lead to more transcription. So next, coming to silencers, in the name itself you can see it can repress or it can prevent the RNA polymerase binding so that it will decrease the transcription, that is our repressor. Next, coming to epigenetics. Epigenetics are the changes which are made to the gene expression without a change in underlying DNA sequence. How to understand this? Without any change in DNA sequence, you are changing the gene expression. That is mainly seen in the case of DNA methylation, histone methylation, acetylation. In this case, we are not changing any size or anything in the sequence of DNA, but we are changing the gene expression. Because of methylation, we are muting the gene. Because of acetylation, we are activating the gene. But actually, we need an enhancer or silencer. Here, the gene expression is changing. But in the case of methylation, acetylation, we are not changing any gene expression, and simply we are leading to the change in the expression of gene. Now let us understand this RNA processing, especially this is in the eukaryotic cell. You can see the functional organization. So the first one which you see is a DNA coding strand. and the second one you see is the pre-mRNA, and next the third one you see the mature mRNA, and finally you see the protein. So first, from the DNA there should be a process called transcription to form pre-mRNA. This pre-mRNA is actually, you can also called as hnRNA. What is hnRNA? It is a heterogeneous nuclear RNA which is modified, and later it can become into mRNA. mRNA means it is a modified or mature mRNA. This can be formed from hnRNA. So for hnRNA we should do some changes, like we should add the cap, that is 5' cap. We can add this by adding 7-methyl guanosine cap, and we can also add a tail called poly-A tail. You can see here this is polyadylation of 3' so this is a 5' on 3' we need to add some tail to the DNA so that it is modified into mature mRNA. And also we need to remove the introns. You can see there is intron, exon, so we need to remove all these introns. That is called splicing. We remove just all the introns and only exons are present, and that is called as mature mRNA. So mature mRNA should have 5' cap, it should have poly-A tail in the 3' end, all the introns should be removed, only exons should be present, then this is called as mature mRNA. And this mature mRNA now can be transported from the nucleus out to the cytosol, and in the cytosol it is translated into proteins. But this mRNA quality control is done by some P bodies called as processing bodies. So there are some P bodies in the cytosol. They process, they see whether everything is matured and perfect. So if something is still not decapped, they just have some decapping enzymes, so they'll remove this cap. And they also have exonucleases which remove the exon, which also remove some exons, they remove the decap, and they also have microRNAs. So the mRNA may be degraded or stored in P body for future translation. So if they need proteins, then these P bodies will store. If they don't need, they can degrade them. And one more thing, poly-A polymerase does not require template. So very important, poly-A tail is formed by poly-A polymerase. It does not require any template. And you can see here the AAU AAA, this one is polyadylation signal. So if there is some mutation in this signal, then that will lead to early degradation of this mRNA prior to translation, that it will not undergo translation and this will undergo degradation. Now let us discuss about RNA polymerases. So RNA polymerases is the one which forms the RNA from DNA. So DNA is converted into RNA now to form some transcription. After transcription, translation, right? So they don't require any primer. RNA polymerase don't require any primer, but they require some transcription factors. So in eukaryotes, RNA polymerase is divided into 3 types: RNA polymerase 1, 2, 3. RNA polymerase 1 forms rRNA, RNA polymerase 2 forms mainly mRNA and also miRNA, snRNA, all these things. RNA polymerase 3 mainly forms the tRNA. So this RNA polymerase don't have any proofreading. If you remember, in DNA you have proofreading from 3' to 5'. In DNA the synthesis is from 5' to 3'. But here there is no proofreading required, but they can initiate the chains. So RNA polymerase 2 mainly opens the DNA at promoter site. The promoter site start up all those cat boxes there. The opening of DNA is first done by RNA polymerase 2. And very important thing, this all RNA polymerase 1, 2, 3 are forming rRNA, mRNA, tRNA, which are very useful. We'll see one of these uses in coming thing. Especially mRNA is very important because this mRNA only will later convert into mature mRNA, and that will go out and translate and form protein. So for example, you have some alpha amantine which is found in some mushroom called Amanita phalloides. This is a death cap mushroom because it inhibits RNA polymerase 2. You can understand, when RNA polymerase 2 is not produced, then the DNA can't be converted into RNA and it can't be transcribed. So this will lead to dysentery, severe hepatotoxicity, and if ingested, it can sometimes also lead to liver failure problem. There is also dactinomycin which inhibits the RNA polymerase in both prokaryotes and eukaryotes. And in prokaryotes, you have only one RNA polymerase, that is RNA polymerase 1, which can only form all three types of RNA, that is rRNA, mRNA, and tRNA, can be formed directly by only RNA polymerase 1 in prokaryotes. Their transcription factor is sigma factor, and here the actinomycin D can inhibit this RNA polymerase 1, and also this rimphamycins can inhibit the RNA polymerase in prokaryote. So there dactinomycin, this mushroom can inhibit. Here rimphamycin and actinomycin can inhibit RNA polymerase 1. Now let us understand very important topic, that is the splicing of pre-mRNA. You know the pre-mRNA should undergo splicing. Splicing is nothing but removal of introns. You can see here the intron is removed and adjoining exons. So this is the process of splicing, but we need to see some important you know RNAs which are involved here. For example, this GU end and AG end is where we need to remove this, because this is an intron. Intron have a GU end and AG end. GU end is on 5' side, AG end is on 3' side. We need to now remove this by means of some proteins like snRNPs. snRNPs are formed by RNA polymerase 2. Here you can see they are forming mRNA, miRNA, and snRNA. So this snRNA along with mRNA form a special structure called spliceosome. So spliceosome is mRNA plus snRNP. You can see here snRNPs plus mRNA form spliceosome. So by means of this spliceosome, you can see here this blue color like structure. Because of this spliceosome, we are able to form a ring like structure, and just we are going to remove this out and mix the two types of exons and form a mature mRNA, because mature mRNA should have only exons, there should be no introns. So if there are some problems in this snRNP, like in diseases like spinal muscular atrophy, there is snRP assembly disorders. Because of decrease in protein like SMN protein. SMN protein is decrease in congenital degeneration of anterior horn of spinal cord, that will lead to symmetric weakness like hypotonia, floppy baby syndrome. And one more thing you need to know is that this snRNPs and snRNAs, both of these are bound to some proteins called Smith proteins. So by binding to the Smith proteins only, they are able to form a spliceosome which cleaves the mRNA. So if there are some antibodies like anti-U1 snRNP antibodies against this, then the spliceosomes can't be formed and we cannot remove the intron, so that will lead to defective formation of proteins, right? This is associated in diseases like SLE, mixed connective tissue disease, rheumatic diseases. In all these diseases, the problem is the snRNP and the spliceosomes are not formed properly, so we can't form a nice proper protein. This shape structure is called larient. It contains introns and some mRNA. mRNA with introns is called larient, and now we remove the larient and finally join the exons and form a mature mRNA. So mature mRNA is required for formation to undergo transcription and finally to form translation and proteins. So if there is defect in snRNP or snmRNA, that will lead to many problems like mixed connective disease, rheumatic disease, etc. So let's wind up this topic by again recalling all the things which are going on. So first, the DNA is present. RNA polymerase 2 will come and split this DNA and convert into RNA. So you have RNA polymerase 1, 2, 3. 1 will form rRNA, 2 will form mRNA, 3 will form tRNA. Especially this DNA should undergo transcription and form hnRNA, and this hnRNA should undergo the process of splicing. That is, we need to remove all the introns. Actually, exons contain the actual genetic information, whereas introns are regulate the gene expression. So introns are also required, but introns are present only inside the cell. Once after they step out of the nucleus, only exons are present. Introns are all removed by means of splicing. So after removal, it will form a pre-mRNA, and after capping and adding a tail and later going into the P bodies, after all those things will form the mature RNA. After mature RNA, it will undergo the translation process and finally form different kinds of protein. So if you alter the process of splicing, then you can also alter the proteins, right? Because if you alter the different varieties of HNRAs, For example, transmembranes are formed, secreted IgG from the same type of protein in different locations. You get different types. You can see tropomyosin variants in muscles, dopamine receptors in brain. There are very different types, right? Host defense evasion by tumor cells. So different types are formed by different alternatives. Let's continue the last part of molecular biochemistry. So that is with tRNA. So actually the tRNA transfers the amino acids to the protein. So let us see the tRNA structure first. tRNA structure. So you can see tRNA is having a structure of a clove leaf shape. So this is actually made up of 75 to 90 nucleotides, and it has a 3' end where you have CCA. CCA are, you can remember them as can carry amino acids. Okay, this is 3' amino acyl end, and opposite to that this is called as anticodon end. Okay, you can see here this opposite one is an anticodon loop. This is attachment site where CCA is present in both eukaryotic and prokaryotic cells. There is a T arm, D arm. Okay, you can see the T arm is having TUC structures. T stands for ribothiamidine, Psi stands for pseudouridine, C stands for cytidine. Actually, this T arm is useful for the tRNA binding with ribosome. So you can see the binding, I have written here binding because T arm tethers tRNA molecule to ribosome. So that's what. Now tRNA's function is to form proteins, right? So first this tRNA should bind to our ribosome so that it can form proteins. Next, coming to the D arm. D arm contains dihydro, you can see a D, that is a dihydrouridine, which is necessary for tRNA recognition by correct amino acyl tRNA synthase. So actually should correct the find out the amino acyl tRNA synthase properly. So it requires this D arms. So D arm allows detection of tRNA by amino acyl tRNA synthase. So next, coming to attachment site. This is also called as attachment site, that is ACC. ACC should now read it from ACC like this. That is from the 3' to 5' end. This is the acceptor site. So ACC, acceptor site. Here hydroxyl end is added. Next, coming to charging. So charging is mainly starting because of this amino acyl tRNA synthase in the presence of ATP. Now it is converting and becoming charged molecule of tRNA to the codon, which is responsible for accuracy of amino acid selection. So amino acyl tRNA synthase matches an amino acid to tRNA so that it can scrutinize the amino acid before and after it binds to tRNA. So something, if it is incorrect amino acid, then the bond is hydrolyzed and a mischarged tRNA will lead to the usual codon but inserts just the wrong amino acids. So that is important here. The amino acid will be wrong if it is wrong type, if it is mischarged, but if it is normally charged, that is amino acylation, if it is normal, then you will get the proper amino acid. So that is about charging. Next, after charging, what happens is, you can see here there is initiation factor which converts the charging into pairing part, that is the codon and anticodon. You can see this is a anticodon and this is the codon sequence of a mRNA. So mRNA to tRNA, that is the transferring of amino acids to protein. So now from the tRNA you can start forming a protein, that is a translation process. So next, coming to start and stop codons, you have here, you can see eukaryotes and prokaryotes, they are different. You can see AUG augments, or you can say it will start the process of protein synthesis. In eukaryotes it will code for methionine, which may be removed before translation is completed. Okay, and in prokaryote it's code for N-formyl methionine, which stimulates neutrophil chemotaxis. Next, mRNA stop codons, you go away, you are gone. These are the mnemonics: UGA, UAA, UAG. Okay, these are the stop codons. Now let us discuss about protein synthesis. Protein synthesis is divided into initiation, elongation, and termination. So initiation of this part is initiation. It is mainly done by ELF. ELF means eukaryotic initiation factor. So there are those factors which identify the 5' cap end of this sequence, and they also assemble the 40S ribosome subunits with the initiated tRNA. You can see initiated tRNA. and also they collect the ribosome from 60 as subunit, and all this assembling requires the GTP, that is the energy GTP is required. GTP is required for gripping and translocation. ATP is required for charging. You can see here ATP is required here for charging of tRNA. GTP is required for gripping and also some going places, that is translocation. An important thing here you need to see, eukaryotes 40s plus 60s ribosome, that is 80s even. Prokaryotes 30s plus 50s, that is 70s prime. So prokaryotes is prime, eukaryotes is even. Now coming to elongation process. So in elongation process, here you can see clearly the amino acyl tRNA binds to the A site. So there are three sites actually, A, P, E. So the first one is amino acyl tRNA binds to the A site, so that it requires an elongation factor and GTP, both are required here. Next, coming to the second stage, here you can see it is mentioned too, that is rRNA ribozyme catalyze peptide bond formation and transfers the growing polypeptide to amino acid in A site. You can see here this is transferring, the growing polypeptide is transferred to the A site of amino acid. Next, coming to the third part, that is the ribosome advances 3 nucleotides towards the 3' end of mRNA, moving peptidyl tRNA to P site. So you can see from A side, now the mRNA is moving peptidyl tRNA to P site. This is translocation. So now the peptidyl part of tRNA moves to the P site. Okay, so here you can see the mnemonic A, that is A for incoming amino acid tRNA, P for accommodating growing peptide, E for empty tRNA as it exists. So elongation factors are actually target for bacterial toxin because they want to prevent the elongation of this tRNA. So like diphtheria, pseudomonas, they attack on the elongation factor. Now coming to the final part, that is termination. You can see that is here where eukaryotic release factors recognize stop codon and they halt the transition. So completed polypeptide is released from ribosome and it requires again GTP, again energy is required to form now the protein. There is one is called as Shine-Dalgarro sequence, where ribosomal binding site in prokaryotic mRNA. Actually, this is a ribosomal binding site in prokaryotic mRNA. It is recognized by 16S RNA in ribosomal subunit and enables the protein synthesis initiation by aligning ribosome with start codon so that the code is read correctly. Now let us see what are post-translational, after translation what are going on. First undergoes trimming, that is we remove the N and C side terminal propeptides from the zymogen to generate some mature proteins like trypsinogen to trypsin. So we just remove N and C side. Next, covalent alterations like many phosphorylation, glycosylation, hydroxylation, methylation, acetylation, ubiquitation, all these are important. Especially hydroxylation is important for collagen synthesis. Next, finally, chaperone proteins. These are intracellular proteins which are involved in facilitating and maintaining protein folding. So we need to fold the protein. For that we need this protein. In heat shock proteins are expressed, but expression may increase with high temperatures and acidic pH and also hypoxia to prevent protein denaturing and misfolding. So by this we complete the molecular part of biochemistry. Next we'll start with cellular part.